Now it's a really true pleasure to welcome Philippe Moreau. I don't need to introduce Philippe, who is really a big leader and pioneer in the field. And I think it's always to take some time to mature more and more data. I don't think there is a lot of, you may think that we are repeating, the monoclonal antibodies data, but I think these are really a true revolution in the field of myeloma, and we need to mature this data. So Philippe will address the case of monoclonal antibodies in myeloma.
So thank you, Mohamed, for this kind introduction. Thank you for this invitation as well, and thank you, Professor Nagler and Thierry, as well, for this invitation. So, while you were speaking about a revolution, Mohamed, and it happened, in fact, with non-Hodgkin's lymphoma, the addition of Rituximab had a huge impact, in fact, to the CHOP regimen, and you know that with a very long follow-up, the overall survival of patients with B-cell lymphoma was highly improved with Rituximab, and Rituximab became, let's say, the backbone of the treatment of non-Hodgkin's lymphoma. And why, in fact? And Rituximab is the drug that is the most widely used in hematology, in fact, because the target CD20 is highly expressed, because there is not only a PFS, but also an overall survival benefit, not only in high grade, but also in follicular lymphoma, also in Burkitt lymphoma, also recently in ALL as well. We know that this drug is not associated with a severe toxicity. It is feasible in frail patients. It is effective in a frontline setting, in the relapse setting, in a maintenance setting. We know that we can retreat patients with Rituximab. We know that there is no long-term toxicity, and now we are using Rituximab sub-Q, so a true revolution for our patients and in our routine practice. So do we have the same, and are we going to reach the same goals, in fact, with monoclonal antibodies in multiple myeloma? So this is the issue, in fact. So you know perfectly this picture, how monoclonal antibodies are acting in myeloma with a direct effect when the antibody is targeting the antigen, with activation of macrophages, with the ADCP activity, with the activation of NK cells, with this ADCC activity, and also with the activation of the complement system to induce the apoptosis of the tumor cells. Well, monoclonal antibodies, in fact, in myeloma have been used for more than almost 20 years, in fact, with several targets. And the first attempt was CD20, in fact, based on the Rituximab story. You know that something like 10 to 15% of the patients are expressing CD20, and the first attempt was a negative one, but the patients were not selected based on CD20 expression. And we looked at that on myeloma expressing CD20 with IFM and only a minor response. That was also tested with CD40, in fact, and we do have data showing that no responses when targeting this antigen that is frequently expressed by the tumor cells. What about CD56, that is also highly expressed on the tumor cells? Single agent, no activity. In combination with Lendex, the response rate was not increased. So a lot of failures, in fact, in the development of monoclonal antibodies. We looked also at IGF-1 receptor as a target. You know that IGF-1 is a molecule that is able to promote the tumor cell survival. And in fact, we tested single agent in combination with bortezomib index and no activity, in fact. So very disappointing results. We looked also at CD138 and similarly disappointing results. So a very long story of failures with monoclonal antibodies. We looked also at IL-6. That is also a very important molecule for the survival of the tumor cells. And you know that Rituximab was associated as well with disappointing results in phase 2, very large phase 2 trials in combination with bortezomib and dexamethasone. and the development of Elotuzumab targeting CS1. CS1 is always expressed by the tumor cells with a limited expression on normal tissues. And Elotuzumab has a dual mode of action with a direct activation of the NK cells and also a direct apoptotic effect on the tumor cells. So you are knowing very well this ADCC mode of action of ELO, and in ex vivo and data and also data on mice models showed that ELO had a synergistic activity with bortezomib, but mostly with lenalidomide. And we know as well that elotuzumab has no single agent activity. So ELO was developed in combination with Lendex in a randomized phase 2 study, and 2 doses of ELO were tested, either 10 or 20 milligrams per kg IV on 73 patients. This is a study schedule with elotuzumab proposed IV once weekly and then every 2 weeks. And the data were published quite recently in the Lancet Hematology. You can see that the median PFS in the relapse setting was 28 months. Lendex alone, that's something like 14, 15 months in median in the relapse setting. So we thought that these data were really outstanding, 28 months in the relapse setting. Very promising results that were the basis of the ELOQUENT-2 study in relapse, comparing Lendex versus Lendex plus elotuzumab on a very high number of patients. And you know that the progression-free survival in the ELO arm was highly improved from 15 to more than 19 months with a hazard ratio of 0.7. The time to next treatment was also improved with the addition of elotuzumab. And importantly, when looking at the toxicity of the triplet combination, very few differences when comparing Lendex, any grade, Lendex grade 3 or 4 versus Lendex plus elotuzumab. So a very safe combination. Lymphopenia was increased, but the number of infections was not increased in the ELO arm of the study. What about infusion reactions? We are dealing with monoclonal antibodies. So with ELO, very few grade 3, only 1% of the patients are experiencing grade 3 toxicity, and mostly at the time of the first infusion. So based on these results, ELO was approved in combination with Lendex, both by the FDA and EMA for the treatment of relapsed patients who have received at least one prior line of treatment. So ELO is currently also developed as part of frontline treatment, and we are awaiting for the results of this phase 3 ELOQUENT-1 study, frontline patients not eligible for stem cell transplantation, Lendex or ELO Lendex, and we are expecting maybe the results at the end of this year at the next ASH meeting. And our colleagues in Germany are also testing ELO in the context of stem cell transplantation. They are proposing RVD stem cell transplantation, RVD consolidation, Lendex maintenance, with or without elotuzumab. So a lot of development of this monoclonal antibody. So to summarize, what we can say about ELO in multiple myeloma. The target is highly expressed. There is a PFS benefit. We don't know any OS benefit right now. It is effective in the relapse setting in high-risk patients. We don't have any severe toxicity. It is feasible in frail patients and mostly in the relapse setting, and we are awaiting for frontline data, maintenance data. We don't know if retreatment is feasible. We don't know about the long-term toxicity, and ELO will not be developed sub-Q. So the other target that is now the most important one is CD38. And you know that we do have 3 monoclonal antibodies that are currently developed. One is a chimeric one, is Rituximab, and 2 fully human, Daraa and MOR202. So what about Daraa? Daraa, you know the mode of action with this CDC, ADCP, ADCC mode of action, an anti-tumor effect, a direct anti-tumor effect. But the group of Amsterdam and Rotterdam in the Netherlands showed nicely that Daraa was also able to suppress some immune regulatory cells, Treg, Breg, MDSC, and when these cells are targeted by Daratumumab, we have an induction of a clonal T-cell expansion and enhancement of the T-cell response that is able to induce apoptosis of the tumor cells as well. So Daraa was developed in Europe in a phase 1-2 study published in the New England Journal of Medicine 1 year and a half ago, the Gen 501 study. The same study was reproduced in the US, the Sirius study, a phase 2 study in very advanced patients. And these two trials, Gen 501, Sirius, were combined and published recently 1 year ago by Dr. Usmani in Blood, the clinical efficacy of Daraa single agent in very advanced patients at the dose of 16 milligrams per kg IV on 148 patients. These patients were very advanced. They were most of them refractory to bortezomib, almost all of them refractory to lenalidomide as well. Many of them were refractory to carfilzomib, to pomalidomide, to alkylators. So true population of unmet medical need. And the results are the following: 30% of response, but also 45% of patients are stabilized, although they were progressing on their last line of treatment and truly refractory to almost all agents available. This is the progression-free survival, 4 months in median, but when looking at the PFS of responders, 15 months in median. On the opposite, patients with a minimal response or stable disease, 3 months median PFS. The overall survival in median was 20 months, and when looking at the survival of responders, you can see a huge impact, and many of them are still alive, more than 2 years following the onset of Daraa. And this subgroup of patients with a stable disease, the median OS is 1 year and a half, so a very important benefit in terms of overall survival. And based on this data, Daraa was approved not only in the US but also in Europe. And the drug is very safe as well. We do have half of the patients experiencing infusion-related reaction, mostly grade 2 and mostly at the time of the first infusion, but very few discontinuation due to this infusion-related reaction. Daraa was immediately tested earlier in the course of the disease, and you have seen this data in combination with bortezomib and dex many times during this meeting. The paper was published last summer, a prospective comparison in 500 patients of VD, bortezomib fixed duration, versus bortezomib dex fixed duration 8 cycles plus Daraa with the primary endpoint of progression-free survival. And you know very well now this huge benefit in terms of PFS. Overall survival data are not mature. We have to wait for a longer follow-up. And the toxicity with the addition of daratumumab to bortezomib and dexamethasone is not increased. So this triplet combination, VD-Daraa, is now approved in the US and in Europe as well. Last October, Lendex plus daratumumab was also reported in the New England Journal of Medicine, a phase 3 randomized study. More than 560 patients were randomized with Lendex until progression or Lendex plus Daraa until progression, the same endpoint, progression-free survival as a primary endpoint. And this best hazard ratio ever reported, 0.37 in the relapse setting for myeloma patients. The overall survival data as well are not mature, and you've heard about these results in terms of MRD negativity, especially at the level of 10 to the minus 5. That is the one that is considered as optimal by the International Myeloma Working Group. You can see that 1/4 of the patients are reaching MRD negativity with Daraa Lendex. as compared with only 5% of the patients with Lendex alone. And this is associated, of course, with a better progression-free survival. You can see this huge impact of MRD negativity in the Lendex arm, but also in the Lendex-Daraa arm, and obviously the number of patients in the Lendex arm reaching MRD negativity is much lower as compared to the number of patients reaching MRD negativity with Daraa plus Lendex. And you've heard also about the results of cytogenetic evaluation with the Daraa Lendex versus Lendex alone. For standard risk patients, a huge difference, but also in high-risk patients, Lendex and Daraa Lendex, a big difference as well. But you see that the curves are not superposable when looking at standard risk versus high risk in the Daraa arm of the study. So Daraa is not able to overcome the poor prognosis status in the relapse setting. So Daraa is now currently developed in the frontline treatment, and Lendex versus Lendex-Daraa, the Maya study, is now closed for enrollment as part of frontline treatment in elderly patients. And we do also have this VMP, one of the standard of care for elderly patients frontline versus VMP-Daraa, that is currently ongoing, the Alcyone study. And you know that Daraa is also tested in a very large study from IFM and from the Hoven group, VTD stem cell transplantation, VTD consolidation, plus or minus Daraa. And Daraa is also tested in the maintenance setting. So you can see that the development of Daraa is very quick, in fact. And Daraa is also tested sub-Q, and we have heard at the last Myeloma Workshop about these early results of the PAVO study. And Daraa was tested at 2 flat doses, 1200 milligrams or 1800 milligrams sub-Q. And very interestingly, the infusion-related reactions were lower with sub-Q as compared with IV. That's very interesting for the patients for sure. And the PK evaluations show that the optimal dose was probably 18 milligrams flat dose, 1800 milligrams sub-Q, very similar to that observed with 16 milligrams per kg IV. So we are awaiting for this possibility of using Daraa sub-Q in the near future. So to summarize Daraa, in fact, activity in myeloma, the target is highly expressed. We do have a PFS and a trend for overall survival for relapse patients. It is effective in the population with high-risk disease. We don't have any severe toxicity. It is feasible in frail patients. And currently the data are positive in the relapse setting, and we have to await for frontline results, maintenance results. We don't know if we can retreat patients with Daraa. We don't know about the long-term toxicity, and there is a development of sub-Q Daraa. Elotuzumab is another monoclonal antibody targeting CD38, currently developed in the relapse setting as well. We've heard about the response rate, single agent at the dose of 10 milligrams per kg, and the results, response rate, are very similar to that of Daratumumab. And we've also heard about recent data of Lendex plus Elotuzumab at the dose of 10 milligrams per kg IV. These data are currently in press in blood and will be very soon reported on 31 patients, and the response rate were rather identical to that of Daraa Lendex, and some patients were Len refractory and they were responding to the triplet combination with Elotuzumab as well. And currently a phase 3 study is ongoing comparing Pomdex plus or minus Elotuzumab. This is a pivotal study for the approval of this monoclonal antibody targeting CD38. This study is ongoing in France and in Europe as well. The third monoclonal antibody targeting CD38 is MOR202, and we have very few data right now available with this third agent. So these monoclonal antibodies are now becoming available and approved in very advanced patients in the relapse setting. So are they going to impact on our current clinical guidelines? And I showed you here the ESMO guidelines that will be published very soon for elderly patients frontline. What are currently the proposal in 2017? VMP, Lendex, or VRD? And what are we going to do in the future based on the ongoing trials looking at monoclonal antibodies? We do have at least 3 randomized studies ongoing in elderly patients, very important ones, and this slide was presented yesterday by Thierry Facon. Lendex plus or minus Ixazomib, a PI, oral proteasome inhibitor, tourmaline study. We have Lendex plus or minus Elotuzumab. We also have Lendex plus or minus Daratumumab, the Maya study. So we can maybe anticipate that in 2019 or 2020, the guidelines will be modified to have this Lendex Daraa, Lendex Elo, and also I mentioned the VMP plus or minus Daraa Alcyone study. So maybe we will have an impact on frontline treatment in the near future. These are the guidelines for young patients currently in 2017, a triplet induction regimen, stem cell transplantation and LEN maintenance, and we know that currently Daraa is tested frontline with VTD. We will also have a VRD plus or minus Daraa study prior to stem cell transplantation, and I mentioned this VRD ELO study that is currently performed in Germany. So we can also anticipate that the guidelines will be modified in 2 years from now with the results of this ongoing study. And in the relapse setting, what are we proposing in 2017? When patients are progressing on Lendex frontline, we can use Daraa VD based on the CASTOR results. When patients are progressing following a bortezomib-based induction, we can use Daraa Lendex based on Pollux. We can use Elo Lendex based on Eloquent. For patients with 2 or more prior lines of treatment, we can use Daratumumab single agent in very advanced patients. And in the future, based on the current ongoing trials, we are going probably to add Pomdex Daraa or Carfilzomib Dex Isatuximab, that is also a very important phase 3 study ongoing. So my conclusions are the following. In fact, we have Lendex Elo approved, and we have also a higher number of possibilities with Daraa single agent in very advanced patients with Lendex or bortezomib dex. And my personal opinion is that Daraa will become in the future the backbone of the treatment of myeloma, not only in the relapse setting but also for frontline treatment of almost all patients with myeloma, very similarly to what Rituximab is currently used for non-Hodgkin's lymphoma. Thank you for your kind attention.